Thank you. In 1787, the Italian anatomist and geologist Paolo Mascagni published a masterpiece. It was a nearly complete map of the human lymphatic system, and he included detailed representations of lymphatic vessels surrounding the brain. Unfortunately, by a stroke of historical misfortune, Mascagni's findings of lymphatic vessels surrounding the brain were dismissed. They were forgotten. They've been ridiculed for the next 200 years until in 2015, these lymphatic vessels in the membrane surrounding the brain were rediscovered. It was a big splash. It was named as one of the top scientific discoveries of 2015. So you have lymphatic vessels in almost your entire body from the periphery up in the membrane surrounding your brain. But, and there is an important but, there are no lymphatic vessels within the brain itself. Now, the lymphatic system plays several purposes, but one of the key purposes is to clear water and metabolic waste from the tissue and out. And considering that your brain uses about 20% of your metabolism, there seems to be a need for the brain to clear metabolic waste. So a big question becomes, what plays the role of the brain's lymphatic system? And the interesting thing is that nobody knows. We really do not know. Now, you may wonder why you should care about your body's and possible brain's lymphatic system. Take a look at this. So this illustration shows so-called amyloid beta plaques in brown and tau tangles in blue in the space around or within brain cells. These clumps of protein aggregations are hallmark features of Alzheimer's disease and also other neurodegenerative diseases like Parkinson's disease. The substances, amyloid beta and tau, are natural. They're natural byproducts of your metabolism, but these aggregations, the fact that they lump together, is not natural. And this is really this hallmark characteristic of these neurodegenerative diseases. We do not know why or how these lump together, and we do not know the precise role of these lumps in the disease or disease progression, but there is a clear correlation between these features and neurodegenerative disease. And one idea is that there might be something about the brain's transport mechanisms that goes wrong and that somehow contributes to this formation of these lumps. We don't know, nobody knows, but our premise, our agenda, my research team agenda, is to try to better understand the mechanisms underlying brain phatics, the interplay between fluid and structure in the brain, in order to long-term better understand neurological or neurodegenerative disease. So this is a lot of things that we don't know about our brain, but what do we know about the brain? And in order to say something about that, let me slip a few slides backwards and give you a crash course in brain anatomy. So the brain parenchyma in brown here is surrounded by membranes. The thickest outermost one, the dura, the thinner middle one, the arachnoid membrane, and the innermost pia membrane. Between the arachnoid and the pia membrane is a space called the subarachnoid space. It's filled with CSF, or cerebrospinal fluid, a water-like fluid, and it's connected to the spinal canal. Blood vessels line the surface of the brain, and they also dive into the brain, both arteries and veins. Now, with that in mind, what are the mechanisms, what are the pathways for solutes to move into or out of the brain? Well, let's first look at this distribution of what we know of observations of tracer in the brain. So a tracer is a biological dye can think of. And here in these experiments, they've injected this dye in the spinal cord, and they're observing using MRI how the tracer spreads in the brain parenchyma. And the tracer will move up along the spinal canal, and it will spread into the brain in the matter of hours, and it will have covered the entire surface of the brain at least in 24 hours. Now, these data originate from the only team in the world that has this as a part of their clinical practice for some cohorts of dementia patients. So this tells you something about how rare these types of data are. And you can also say something about the temporal sparsity of these data. Now, in animals, you can explore a bit more, and you can zoom in, and it seems that the tracer moves along the blood vessels on the brain surface and then dives in, and following the blood vessels, dive further into the brain. This is not a static process. It turns out that this tracer influx is faster with sleep. So sleeping mice transport tracer faster than awake mice. Exercise also has a positive effect, but also alcohol has a positive effect. Note, however, that a moderate amount of alcohol has a positive effect, but not a large amount. This is a field where some things are known, but very much is not known. This is a field, it's a very active, very exciting field. There's a lot of controversy, conflicting findings, and big open questions. We're wondering, what are the mechanisms underlying this influx and clearance from the brain and its environment? Are the forces and spaces sufficient in order to create an effective transport? And how do these processes relate to these diseases, if at all? We are trying to use computational modeling and simulation in order to address some of these questions, but this is really a frontier field. So the current state, what we really can contribute with is to evaluate biological hypotheses using physics-based insight, or we can evaluate mechanisms and predict distributions. And when I say we, I mean our team here at Simla, our clinical collaborators at Oslo University Hospital, our collaborators in Oxford and beyond, and in particular, my former students, Matteo Crocci, Marius Kauseman, Eleanor Piersanti, and Vegard Vinja, who should receive much of the credit for the work presented here today. So with that, let's dive in. So as we observed, we saw that if you inject this tracer in the cerebrospinal fluid, it will distribute in the entire brain in the matter of hours and days. The medical doctors claim that it's unlikely that diffusion alone explains the brain-wide distribution. Now, this is a claim that we can investigate, very easily investigate using computational modeling and simulation. So what we did was that we considered families of models. So we extracted the movement of the tracer in the subarachnoid space, set that as a boundary condition for convection diffusion reaction simulations of tracer transport in the brain. And importantly, we try to evaluate the uncertainty. There's a large uncertainty in the diffusion coefficients. There's large uncertainty in potential velocities. So we accounted for the velocity and ran a bunch of Monte Carlo simulations. We computed clinically interesting quantities of interest, such as the average amount of tracer in the gray matter, just the outermost region, or the total amount in the white matter, the deeper regions. We first just looked at what would be the tracer evolution if it only moved by diffusion. And what we found here was not very surprising. It moves upwards, surrounds the cavity, and then starts to move inwards into the brain in the right time scale in the course of hours, days. We found that the expected amount of tracer in the gray matter peaks around 15 hours, which is consistent with the clinical observations. But we found that the expected amount of tracer in the white matter would still be increasing after 24 hours, which is not clear, in agreement with the clinical observations. So this indicates that there may be some additional mechanism at play, but it does not play a big role in the gray matter. what we importantly found was that the uncertainty in the diffusion coefficient really led to substantial variation in all outputs. That's really a dominant source of uncertainty. Now, with this in mind, there may be a convective velocity at play. We went further. And what we wanted to do was to investigate one of the leading hypotheses on convective flow in the brain. This hypothesis is called the glymphatic theory or glymphatic system, and it essentially compares the brain to the city of Venice, a system perforated by channels leading to more effective transport. So the glymphatic theory claims there's a bulk flow on the arterial side, a bulk flow through the extracellular space, and a bulk flow of cerebrospinal fluid out on the perivenous side. Again, we set out to try to model this on a macroscopic scale. And we modeled this by setting some conditions for what such a velocity field would look like. It should conserve mass. It had a randomness. It should represent the typical distance between arteries and venules, and so on. So this way, we could represent this glymphatic velocity in a velocity model. What we then did find was that such a glymphatic velocity would not enhance transport at all, not into any region. So the glymphatic system as formulated here would not lead to enhanced tracer transport. Therefore, we turn to further refinements. We try to see, okay, what if we also have a global directionality associated with the velocity? Maybe a directionality from below and upwards in alignment with the pulse propagation of the cardiac and respiratory pulse. This is what I call VV2 here, that we also used as a velocity field in our convection modeling. In this case, we did observe enhanced tracer transport, moderately so. So to conclude, a global directionality is important for a velocity field to enhance transport in the brain. And this is insight that we can give straight back to the experimentalists and the clinicians to refine their theories. Now let's dig deeper in order to try to understand how these velocities and the interplay in the brain can work. The brain is a surprisingly soft material. It's also rheologically complex. But at the timescales and spatial scales that we're considering, from second up to days and at the whole organ level, it makes sense to look at the brain as a poroelastic material. At this scale, you can really look at the brain as an elastic structure that's permeated by multiple fluid networks, by the blood vessels, by cellular networks, extracellular space, and possibly by perivascular space. Such materials can be modeled via so-called multiple network poroelastic theory, MPET. The core idea is that you have a structure that's permeated by different types of fluid networks, and then you homogenize. So this is a model where all fluid networks exist everywhere, and the tissue also exists everywhere. The equations, the multiple network poroelasticity equations, are listed here, and we're interested in finding the displacement U and a set of J network pressures, Pj. Here J can be 1 to 10, such that balance of momentum and balance of mass holds. We also assume that there's fluid exchange between networks, driven by pressure difference and a Darcy flux model in each network. In the case where J is 1, we have the familiar Biot equations. In the case J is 2, we have Baring-Blatt-Biot. And what's really fascinating is from a numerical point of view, is that there are so many interesting regimes here. The case lambda becomes big, it's the incompressible case. Kj is small in the low permeability case. And both of these cases are super relevant in the brain. At the same time, these interesting regimes may easily be numerically challenging. For instance, this nearly incompressible regime where lambda is big, their numerical approximations are prone to locking or to see a loss of convergence. So considering that the simulations of the brain parenchyma itself is at the core of our interest, this is something that we have to resolve. So we first set out to try to see what can we do in order to have more robust finite element approximations of these equations, where you would regain improved convergence rate. So we were inspired by previous work by Lee, Martel, and Winter for Biot's equations, and also previous work from linear elasticity. So we introduced a new variable. We introduced the total pressure, which is the sum of the solid and the fluid pressures. And by introducing this additional pressure variable, we could transform this system in terms of U and P into a system in terms of U, P0, and P. And the key point is that when lambda gets big, you will have a block in the original system that gets very big, but you will have blocks in this revised system that get very small. And it turns out that this lower system is well posed even in the increstable limit. And we can show this theoretically, and we can show it practically, that now, using this total pressure formulation, we regain the convergence. So we're using this system in order to, and this discretization, as a component of full brain and brain environment simulations. And particularly, we want to understand in detail the mechanisms for how brain pulsates in response to arterial blood influx. And of course, we're now in a clinical setting, so recall that we'll be talking about millimeters of mercury, and 1 millimeter of mercury is around 100 Pascal. So the brain does absolutely not exist alone. It lives in an environment. It's surrounded by the cerebral spinal fluid, and the dynamics of the brain is really an intricate interplay between the blood, the CSF, and the compliance of the tissue. The arterial blood inflow is something that can be measured non-invasively. So here one do have average, but still good data on arterial inflow. There's less data on intracranial pressures. Some things are known about the intracranial pressure in time. It's known that it pulsates in sync with the cardiac and respiratory rhythms and the typical amplitude. But in order to measure the intracranial pressure, this is a heavily invasive procedure. Whether there exists any pressure differences within the parenchyma has been a big question for many years. We recently published a study where we took advantage of some extraordinary data from our clinical collaborators, having simultaneous pressure recordings into different locations in the brain. And there we could identify a pulsatile pressure gradient with a peak amplitude of 1 to 2 millimeters of mercury per meter in a spatial gradient. So that's the order of magnitude we're talking about. So the question becomes, can we now link these dots with what we know about arterial influx? How would the predicted brain pulsatility look like, and how does it compare to the sparse clinical observations that do exist? So we're now solving the MPET-1 or Biot's equations in the brain for the displacement U and the fluid pressure P in the brain. And then these are coupled to time-dependent Stokes equations in the surrounding cerebrospinal space. This is just an illustration. Here you have the MRI-generated mesh. All of this is work by Marius Kausmann, presented in his thesis, Cardiac-Induced Pulsatility of the Human Brain. We have interface conditions between the fluid and the structure, boundary conditions on the boundaries, and what drives the system is an influx of blood into the as a source in Biot's equations following this time form. So this is inflow versus time. Let's see what happens. So as blood flows into the brain is pushed downwards, and you will see displacements that you move downwards and backwards. These are 3 cycles that you see here. The magnitude of the displacement around 0.1 millimeter. Very little data exists on brain displacement, but we think that this is in a plausible range. Similarly, we can also look at the simulated and induced flow of CSF in response to these changes of the brain. We observe that there will be an outflux of CSF from the cranium down into the spinal cord. It will move up into the fluid-filled ventricles inside the brain, and then return. And this will be a rather complex flow patterns. As regard to the pressure, we observe that we get a nearly spatially constant pressure increase, but that you do get these pressure variations that seem in time that seems to be in agreement with clinical observations. So to conclude, we're able to do these fully coupled simulations linking blood flow measurements with the very hard to obtain intracranial pressure and displacement measurements. They compare well with the clinical observations and really offers an unprecedented detail for understanding these mechanisms and also individual variability. So with that, let me thank my collaborators and you for the attention.